The beginning of the action potential graph is known as the resting state. In the resting state, most of the voltage-gated sodium ion channels are shut closed, and some potassium ion channels are open. The resting state is maintained where the interior of the membrane is primarily negative and the exterior matrix is positive.
The second step is called depolarization. Depolarization is a change in a cell's membrane potential, such as the inside of the membrane is made less negative relative to the outside. So the outside, in our example, is more positive than the inside, which is more negative. And then in the graph, you can see that the membrane potentials increases a little bit like that. And then in this step, a stimulus opens some sodium channels, which are these things, Na plus channel. And then the Na plus ions inflow through those channels and depolarizes the membrane. So then say this is a little Na molecule, it goes in and it depolarizes the membrane. And then another sodium ion, it goes in and it depolarizes the membrane. And then if the depolarization reaches the threshold, it triggers an action potential.
The third step is rising phase of action potential. Action potential is a rapid change in the membrane potential of a cell that's caused by stimulus that opens or closes the sodium and potassium ion channels. And threshold is the potential that a cell membrane must reach for an action potential to be initiated. So in this phase, the membrane potential increases up to the action potential, and depolarization opens most sodium channels, but the potassium channels remain closed, and the sodium influx makes the inside of the membrane positive.
The fourth step in the process is called the falling phase of action potential, in which most of the sodium channels become inactivated, which blocks the sodium inflow. Most potassium channels actually open, which permits the potassium outflow, which makes the inside of the cell negative again. So this channel gets blocked, but this channel is still open. So that's why it repolarizes, because now there's a gradient again. And on the action potential graph, it looks like this, because the membrane potential goes back down again.
The next step is called undershoot. The action potential graph, it dips down and then it levels out, so then the signal can't be reversed. Moreover, there is repolarization in this step, which basically means that the membrane repolarizes. Bam, bam. Also in this step, there is a refractory period. This is the downtime following the action potential when a second action potential cannot be initiated, called the refractory period. So this is the refractory period, since the signal is unable to be initiated.
Lastly, after undershoot occurs, it returns to resting potential, which is represented by a straight line on the graph.
This is the parts of a neuron. So up here we have these branches called dendrites. And dendrites are branched extensions that receive a signal from other neurons. Over here is the nucleus, which is the brain of the cell. This is the junction between the axon and the cell, and it is called an axon hillock. This part of the cell is where the signal is generated for the axon to take it and travel to a different cell. At the end, there is something called a synapse, which is where the information is traveled at a junction known as a synaptic terminal. In synaptic terminals, there are messengers called neurotransmitters that send the signal from the presynaptic cell to the postsynaptic recipient cell. There are inhibitory neurotransmitters that hyperpolarize the membrane and excitatory neurotransmitters that depolarize the membrane. On the axon itself, there are two parts. These blocks are called Schwann cells, and the gaps are called nodes of Ranvier. In a myelinated axon, saltatory conduction occurs and the channel hits the node, depolarizes, creates another action potential, jumps up and goes to the next section. Another component is called the synaptic vesicles. These are membrane compartments that house the neurotransmitters in them. The neurotransmitters then come to the postsynaptic membrane and diffuses on the gap between the two cells known as the synaptic cleft. From there, these neurotransmitters can exit the vesicles and go through some ligand-gated ion channels.